First of all, I thought that was an amazing combination of epidemiology, biotechnology, history, politics, touching on mathematical modeling, finance, and raising some very alarming prospects, but at the same time, some practical recipes for what we might do about some of this. So it's very nice to end up with a talk that doesn't just rearrange the deck chairs, which is something I think we may be illustrating shortly on stage. Just as a practical demo, you understand. I would like to introduce then, I'd like to do things. I'd like to thank Professor Adrian Hill, and could we just give him another round of applause? And I'd like to introduce the chair of the panel, Alice Thompson, who's a columnist and interviewer at The Times. Alice, over to you.
I'm going to introduce all the panel, and then after that, we're going to have a couple of questions between us, and then I'm going to open it up to the floor, because you've probably got far more interesting questions than a journalist can ever provide, particularly anyone with a scientific background. I thought that was the most extraordinarily optimistic speech about a very deadly period and a very deadly disease. And it is true, as Professor Hill said, the question that journalists always ask is how long? You know, are you going to get it done? How long are you going to take? Why can't we have it now? But I don't think anyone expected it to be 10 months. I mean, it was just extraordinary for all of us, and I think it is an incredibly optimistic story, but then there are other sides to it that we should explore now, the sort of more pessimistic sides. So the first person I wanted to talk to and introduce and will come up on stage is Nicola Blackwood, who is the MP and is also the chair of the House of Commons Science and Technology Committee. And she has launched her own inquiry into lessons the UK can learn about the Ebola crisis. And we have Dr. Johan von Hoof, who is up there in lights, and is the Global Therapeutic Head of Infectious Diseases and Vaccines at Johnson & Johnson. You can sit anywhere you want, I think. And Professor Chris Whitty, who is Professor of Public and International Health at the London School of Hygiene and Tropical Medicine, who has acted as Chief Scientific Advisor for the Departments of Health and International Development. So really, Professor Hill first, probably we should ask you. What I really wanted to ask is why, first of all, why we can't build a facility. Now you've managed to produce this vaccine. Why can't we build a facility? Why do we have to rely on Belgium, which isn't any more advanced than we are in any way, I assume, and has fewer people and seems a rather extraordinary place to be able to do it when we can't.
I picked Belgium as an example of a country that has a lot of vaccine manufacturing. I'm not clear that we're relying on Belgium or whether Belgium's made any commitment to us. But the real problem is cost. These facilities are pretty expensive to build. We're talking about over £100 million, I imagine, depending on the scale of the facility that you would want. But then that, in a way, is the smaller problem. The bigger problem is keeping them running. These activities of good manufacturing practice are extremely stringently regulated. There's an industry code of practice which is crucial to maintain the safety of these vaccines, and it's just very expensive to keep these facilities running. So if you made one and made, your 60 million doses of Ebola vaccine, what do you do next? These are very specialized staff that you need to hire to be able to do these and sophisticated procedures. So you need a business case not just for building the facility, but for running it. And ideally, you need a versatile facility that could make other vaccines when they're not making outbreak pathogen vaccines. It would help if you were making vaccines of a similar or related type for different disease indications. But expert committees in this country have looked at this before and balked at the cost. And it's more to do with running costs than capital cost.
And Nicola, wouldn't it be a good idea if we became a world centre at this? And we've already produced so much of the research and we're doing so well at Oxford, and it seems to be very much something that Britain's very good at, and we led the way in Ebola. Wouldn't it be a good idea to set up?
Yeah, I don't think I'm on. It's important to make the point we are world leaders already in immunology and epidemiology, and we are brought in to advise around the world. And clearly, we were at the front of the race when it came to developing the Ebola vaccines, and we're already there with Zika as well. But one of the recommendations that we made in our report into Ebola was that while launching the 195 million Fleming Fund to try and kickstart research into emerging infectious diseases and make sure that it was focused in the right direction is a good start, we felt that the government needed to actually target some of that research more effectively and demonstrate to the market that they were in it for the long run. And we recommended an emerging infectious diseases strategy, which demonstrated quite clearly where those priorities were, maybe focusing down to either transmission models or some of the recommendations which you've just heard from Professor Hill, so that not only could you target research, but also industry could look at that and go, okay, the government is in this for the long run and we can start investing there, and you would attract inward investment as well as research and foundation investment and so on to those particular target areas which we want to become world leaders in.
And Johan, you had actually started, hadn't you, your company doing the Ebola virus? You'd started researching it, so you'd got quite a long way down the line. Why hadn't you got further, do you think?
Well, there are many reasons for that. Actually, you should realize, as was mentioned, that we did it in context of the bioterrorism threat in the US. We were doing this in collaboration with NIH. And there are several viral viruses that circulate. So we actually are aiming, we were working on a trivalent vaccine that was supposed to go in the clinic now in 2016. But then with the emergency arising in Western Africa, we decided really to focus on the monovalent one, the Zaire strain. And that's how we could accelerate it. But the point was made before, there's 10 years of research that actually was needed to come to a model, to a vaccine candidate that's really effective in an animal model. With Zika, for instance, there's nothing of that kind today, and that's one of the big differences.
And do you hope that with Zika we'll be able to speed up the whole process as they did with Ebola or not? Or do you think it's too dangerous in many cases to?
So I think there are two separate things to say. And the first is the scientific question. Adrian put an 18-month shortest possible time. I think that's extraordinarily optimistic, and it would require multiple things to go right, even if we went through at an absolutely breakneck speed. So I don't want people to go away thinking that's when it's going to arrive. But I think there's a very big difference between a virus which is killing somewhere between 70 and 90% of the people who get it, where actually you have got to take some risks. And that's what actually happened with Ebola, with a vaccine that had been quite well characterized in animals already, and a virus like Zika, where anyone who's non-pregnant, the risk of serious harm is extremely low. It's not zero, but it is very low. It is only in pregnant women that there is a significant risk. And therefore, if you shortcut the system and then ended up with a vaccine that was actually in some people harmful, you would really regret that. So really shortcutting things was possible in Ebola because of the urgency of the situation. Zika is a very serious problem. There's a lot of effort going in to go through as fast as possible, but cutting corners in the way we did with Ebola probably would not be sensible.
And with Zika, would you then immunise all the women and not the men, or how would it work very differently?
Well, it depends on what you're trying to do. I mean, what you clearly need to do in the first instance is to try and protect women before they get to childbearing age. So it is likely that one of the target groups would be rather like rubella vaccination used to be in girls and women before they get to the point they have their families. But obviously in the longer run, what you want to do is to control these epidemics. And for that, you need vaccination of the whole population. So we're probably going to need to be able to do both of those. But clearly, what you don't want to do is have a vaccine that's dangerous.
All right. And if you were going for Zika, what other ones would you go for? Aidan, is there a sort of top 5 list of vaccines that you now want to produce and think should be produced of the most dangerous possibilities of
I should probably refer
endemics?
that question to Chris, because he chairs a committee that was tasked with doing exactly that for the UK Vaccines Network. The good news is that several people around the world have tried to come up with this list, and there's a great deal of similarity between the lists that appear. So
is your list just for people in Britain then, or is it's a worldwide list?
So this is a list looking at worldwide potential epidemic threats, because the UK government has taken a very serious view that it has a global responsibility to try and tackle these epidemic threats. To be clear, for some epidemics, a vaccine is actually probably not the key thing. And I think people always leap from epidemic to vaccine. Look, for example, at HIV, now in retreat, fortunately, slow retreat. Vaccine has not played any part of that. In this particular outbreak in Ebola, although a vaccine would have been enormously helpful and will be enormously helpful, actually it was controlled by classical epidemiological methods. So the first thing is, for some situations, a vaccine isn't the right answer. But there is quite a clear list of what Adrian in one of his slides called the known knowns, and he gave a good list of those, of which the MERS virus that's circulating in the Middle East, Zika now, and SARS and Ebola viruses, Marburg and Lassa, I think most people would agree. There are a number of others that people would sort of say, where should it be on the list? But most people would agree with those. And I think the general view is for most of these, getting a vaccine is a seriously realistic scientific possibility. There are some diseases which, like HIV and malaria, which are intrinsically a lot more difficult. And I think we just got to be realistic about that. But going from a list of viruses to actually achieving a vaccine, there are many, many steps. So I don't want to jump all the way across. But that list, I think, is achievable over time. And I think there's broad international agreement this is the right list.
And for Johnson & Johnson, how do you decide which ones to go for? Is it purely done on financial basis or on
Certainly not.
who
I
asks?
think it's really a combination of factors where, on one hand, there has to be a huge unmet medical need. And on the other hand, it must be feasible technically in the science. And then, indeed, it must have a viable business case. But the last one is not the only determining factor. For instance, for the HIV vaccine, we know that there have been many failures in the past. At the same time, thanks to the HIV trial that was successful in Thailand a few years ago, even though only at 30%, but at least it was shown that efficacy can be achieved, protection can be achieved. At the same time, correlates that, or biomarkers that correlate with protection were identified. So this actually gave us the courage to continue, because now you know that A, it's feasible scientifically, and B, you do have surrogate markers that you can actually follow in your vaccinees early onwards in your trials to see if you are successful in fine-tuning your vaccine. And that's what we do here.
And with governments, how involved do you think the government should be in all this? Is it vital to us? Is it vital for our safety or defence? Or
Well, it's vital on both of those levels, but I think more than that, one of the things which the government can do is ensure that we're prepared in terms of emergency response, so that there isn't a delay. One of the particular problems which emerged in the inquiry which we held was that we weren't research ready in some areas. So we've heard a lot about vaccines today, but of course there is also the opportunity to do research into therapeutic trials and diagnostics, because if you can't diagnose which patients have the relevant virus, it causes all sorts of problems. With Zika, we currently have a problem that there isn't 100% diagnosis at the moment, and so that's causing all sorts of problems. And so what the researchers told us and the clinicians told us was that because there wasn't established protocols to work out what the research priorities were in an emergency scenario, it was actually quite difficult to make sure that the right therapeutic trials, the right diagnostic trials happened early enough in the outbreak. And in these kinds of outbreak scenarios, even a delay of a couple of weeks costs many thousands of lives. An LSE study found that the placement of therapeutic beds, which the UK had put in place, saved 56,000 lives. But if they'd been put in place just 1 month earlier, a further 12,000 lives would have been saved. And so thinking about these decisions and priorities ahead of time as much as possible, and thinking about, for example, the ethics of field trials for vaccines or for therapeutics with Zika. Are you going to test the vaccine on pregnant women or not, and what are the ethics around that? Will save vital time, and it will save lives.
Perfect. Now, are there any questions from the audience? And we've already got one there. You might need to wait for a microphone, though, and stand up. Perfect.
Isn't one problem with all these diseases that we don't know what epidemic is coming next and when it's coming? Should we not devote more attention to detecting the warning signs, working out what is most likely to happen next? Were there any warning signs of the Ebola epidemic 2 years ago that we missed?
Adrian, what do you...
Yeah, so let me just say that looking back over the last 15 years, of the viruses on that list, all but one was known about, which sounds good, that we aren't getting many unknowns coming, causing very serious outbreaks. But the bad news is that the ones that we have had outbreaks from are from a list of many hundreds. So if you just look at a textbook list of all the flaviviruses, bunyaviruses, and so on that are around, have been described, conceivably could cause an outbreak, you have a big problem in making vaccines for hundreds of those pathogens, and that's just probably not going to happen. So your question is really to do with surveillance. And certainly in response to the Ebola outbreak, there's been a lot more investment in surveillance. This has been improving quite dramatically globally over the last 10 or 15 years after outbreaks of swine flu, avian flu, and so on. We have much better surveillance in Africa now than we've ever had before. It doesn't mean that an outbreak can't happen very rapidly, so rapidly that you don't really have time to respond. It's a big world out there. So I'm not sure that will give you absolute reassurance. And in the case of Ebola, yes, you can trace it back to a case perhaps at the end of 2013 that people might have responded more quickly to. But you know, it's hard because every week, if you look at the reports of cases that are emerging of little-known pathogens, there are a lot of tiny outbreaks, and it's hard to know what's going to really take off. But Chris, you think more about this than I do.
So Chris, are there indicators that make it clearer why it will take off? So why it might take off, say, in Liberia or Guinea, or rather than in another African country?
Well, I think that, I mean, just to answer that, the first question, then straight onto that one, I mean, this was not a failure of surveillance. Let's be clear. We knew this was an Ebola outbreak from relatively early on. It was, in early stages, a failure of imagination that this could take off, and in later stages, a failure of action. Although I want to be clear, I do not consider that was on the fault of the UK government. I think this is an international problem. But the reason we had the failure of imagination is Ebola had outbreaks multiple times previously, and they'd all gone away with public health measures so quickly it would probably have been impossible to intervene with a vaccine, even had a vaccine been there in reality. What was different here is the Ebola got into a society which was both more crowded physically and also had a number of behavioral characteristics that probably made it easier for the virus to be transmitted, including particular burial rites.
Was that very difficult to change?
Yeah, and in a sense, it's not... Burial rites are a central part of any society, and we would not have been able to predict that these particular burial rites were a major risk in West Africa until it happened. At the time it did happen, people did change them, but relatively temporarily. How you bury your family is a very important thing for any family anywhere, and people will have probably reverted back to previous behaviours because that is the traditional way it's done.
Any other questions? Right at the back there. Do you want to say where you're from or who you are, very briefly?
That's working. My name is David. I'm from Lillian Bailey School. My question is to all the panel is, why is it that Britain, such a wealthy country, one of the wealthiest countries in the world, doesn't have many vaccine capacities compared to Belgium to tackle future pathogen outbreaks?
Nicola.
Well, I mean, I think that this is a historical issue to do with just the way in which the farmer community has grown up. But the question I think we need to ask ourselves is, what are we going to do about it now? And the government has already taken some quite significant steps following the Ebola outbreak, investing 195 million in what's called the Fleming Fund, in order to stimulate not only vaccine research but also research in emerging infectious diseases. And I think the question that my committee has asked is how are we going to make the most of that research and focus it on the greatest risks. And that's why we looked at the National Risk Register, which has identified emerging infectious diseases as one of the top risks to the UK since 2008. Am I right, Chris?
I think so.
Yes, since 2008. But Ebola didn't make it onto there until 2015. And our judgment was that while the National Risk Register, the public version of it, is helpful for researchers and for industry to help them understand that this is something that the government takes seriously and will put money into, actually we need something which is a little bit more focused, shows a little bit more determination and political will on the part of the government. That's why we recommend an emerging infectious diseases strategy which identifies key threats, and it could be a list of the individual diseases, or it could be the transmission routes, and this would therefore channel match funding, whether it's from foundations or from universities, and it would direct researchers and industry into focusing on those areas of research. This is what we've seen in other strategies which the government has introduced, and it is an effective way to make sure that we become both more research ready, but also have the right infrastructure in place so that we are prepared for future outbreaks.
Any other questions? Right at the front.
Work for the pharmaceutical industry for a small company called Lambda Therapeutic. My question is related to the fact that the Ebola virus demonstrated an unprecedented, dramatic shortening of the clinical trial pathway. And I wonder whether this might herald a change for the rest of clinical development, which, as we know, is a very prolonged and time-consuming and administratively burdened pathway and process.
Johan, that must be a double-edged sword, because actually if it speeds up too fast and you don't have enough trials, then you worry.
I think it's a very good question. I see several elements in the answer. The first one is that although it certainly went very fast, we should not forget that we all agreed that at no moment we should take shortcuts that take the vaccinee at risk in terms of safety. But what, for instance, was done in contrast to what you do with a classical development is that once you have the safety data, once you know that the vaccine can be given, does not have side effects, you could go into your next step. Why classically you would actually wait till that study is completely over before doing the next thing. And there are downsides to that as well, because you have less information about the immunogenicity and so on, and certainly it's less optimal than doing it sequentially. In addition, we also all agree that this is in a very, very specific risk-benefit setting. It's a very deadly disease, and there in general, the risks that you can take, even if you don't know them exactly, but the theoretical risk you can take is somewhat higher than if it's a not deadly disease, because you know there is no alternative. For instance, there was a debate, and you have mentioned it, about the reactogenicity that was observed with the VSV vaccine, which probably in a prophylactic setting for the general population would not have been acceptable. But it was generally agreed, and I think it was the right decision, that in this particular setting, where indeed it can be used for an outbreak, that you could continue. So I do think indeed that we could learn some lessons from it, and certainly with regulators we could look at, okay, well, have we learned from it and what could be extrapolated to what setting? But I also would caution us to think that you can do it every single time. In addition, also, it generates a lot of extra cost that would come on top of the enormous cost already now of research. So a good question, but I think it's difficult to extrapolate it to other vaccine trials and certainly to therapeutic trials.
Any other questions?
Hi, I am a science student, massive interest in immunology. Professor Hill, you've mentioned that there is an alternative route for licensing vaccines, which doesn't require for active epidemics to be around. So why are there still no licensed vaccines if there is this alternative route?
So for Ebola in particular,
Yeah.
vaccines are going to be licensed, and that is still in progress. So as I pointed out, even for the one vaccine that was tested in phase 3, the VSV, phase 1 had been done, phase 3 was done in Guinea, but a lot more safety data needs to be collected on that particular vaccine before you can complete the file, if you like, of all the registration documents you would have to submit to demonstrate that that vaccine is suitable for licensure. To take one simple example, there's very little in the public domain about how long the immune response lasts. That data must be being collected. It will be needed for registration. So that's the answer for Ebola. I should make clear that even though that route exists, it has very seldom, if ever, been used. So it's there specifically for this group of pathogens where you might not be able to demonstrate efficacy. There's often the rider when you get registration that you have to then go and do trials when there is an outbreak to show that it actually does work. So it's not an easy route, and my sense is it's not a route that pharma particularly like.
Actually, we don't. The reason is it sounds easy on paper, but once you start looking into the details, it is really very challenging. And one of the reasons also is that this rule was originally written for therapeutic drugs, for small molecules, where comparison between animal exposure to the drug and human exposure is much easier to calculate and to analyze than comparing the immune response in animals versus the immune response in humans. And to be honest, and this is not to criticize the regulators, they also struggle with it, and we are in discussion with FDA on how best to interpret this. I think we are evolving towards a situation where we agree that these animal data learn us that the individual can be protected, that they tell us something about the mechanism of protection, that antibodies are important, that cell-mediated immunity is important, but most likely it will be very difficult to deduct any threshold from these animal data because you just don't know how that animal model behaves as compared to the human model. And to the point that you made, the animal model suggests that you need huge amounts of antibodies to be protected, while the rig vaccination trial seems to suggest, if the data are correct, that a low level of antibody can be protective. So I think we are evolving now into a situation where the regulators, especially FDA, will expect us to use these animal data to actually demonstrate that protection is possible, to analyze it, look for the mechanism of action, but then they come to the conclusion that based on all of this, it's highly likely to be of benefit in the vaccinees, and then we will have to commit to do immediately an effectiveness study if it's rolled out during an outbreak, and that's probably where it will end up.
Right. Thank you very much to the panel, and also to Professor Hill. That's absolutely fascinating and very optimistic in the end, I think.
So there are many people have worked hard to make this evening happen. The team led by Margaret Unsley has put an enormous amount of work into this. I'd like to thank them. I'd absolutely like to thank the panel once more. It's very good of you to give up your evenings and be here and to entertain and inform. I would like to thank the audience, you, and because I'm an experimental psychologist, I know that the only effective way of thanking people is to give them a reward. So there will be one. You all get invited to drinks, and the ushers will lead you there in just a moment. We're going to, of course, there's always a delay of reinforcement, makes it more effective. We're going to retain the panel here for a few minutes while we send you off so that we can strip them of their valuable microphones and other things like that. And then we hope to join you, we will join you in the reception, so you might have another chance to talk to people again. But the last thing to do, obviously, is to repeat the thanks to Professor Adrian Hill for an absolutely wonderful evening.